genetics. Dog begets dog, cow begets cow, and mango trees bear mango fruits only. This is called heredity, and genetics is the science which deals with the study on heredity. Gregor Johann Mendel, an Austrian monk, is recognized as the father of genetics. He laid down the foundation of modern genetics, that inheritance depends upon discrete units of inheritance called genes, and he deduced the basic principles of heredity. Human genetics studies inheritance in human beings. There are ideal methods of studying human genetics. Collection of pedigrees of distinctive traits provided the basic data on the study of human genetics. A pedigree is a diagram showing the ancestral relationship and transmission of genetic traits over several generations in a family. Analysis of pedigrees of different traits enables the geneticist to establish the mode of inheritance of the traits. Intermarriages between different human populations have been taking place since the dawn of humankind. Such type of interracial marriage is another good source of information for human geneticists. Advancement in the field of cytogenetics and molecular genetics has made tremendous breakthrough in human genetics during the last 5 decades. Chromosome. The chromosomes are thread-like structures present in the nucleus of a cell. In Greek words, chroma means color and soma means body. They are easily stained when a special dye is added. The chromosomes are chain of genes, which serves to convey inherited traits from one generation to another. Several studies confirm that there are 46 chromosomes in men, that is 23 pairs, of which 22 pairs are autosomes and the remaining 1 pair of sex chromosomes. The 22 pairs of autosomes are said to be homologous, for the pairing chromosomes are similar to one another. The sex chromosomes are of 2 types, denoted as X and Y. In men, normal chromosomal complement is 22 pairs of autosomes plus XY, while in the female, it is 22 pairs of autosomes plus XX. Females all the time produce only one type of egg containing haploid set of 22 autosomes plus an X, while males produce 2 types of sperms containing either 22 autosomes plus X or 22 autosomes plus Y. Sex of fetus depends upon the chance fertilization of an egg with a sperm of either 22 plus X or 22 plus Y. The karyotype, that is the number and types of chromosomes in a cell. The karyotypes of men is now completely delineated. The 46 chromosomes have been classified into 7 major morphological groups, such as A, B, C, D, E, F, G. The chromosomes are numbered according to size. Number 1 is the longest and 22nd is the shortest. Genes. The 46 chromosomes per cell are made up of thousands of tiny pieces are called DNA, that is deoxyribonucleic acid. Each piece of DNA is a tiny leather-like structure. The chemicals involved are simple sugars, phosphates, and nitrogen bases embedded in a protein matrix. Each of the single building units of DNA is called a nucleotide. It is formed by the union of a phosphoric acid molecule and a kind of sugar, that is deoxyribose, a molecule of nitrogen based, there are two classes of nitrogen bases, purine base and pyrimidine base. Purine base is of two types. They are adenine and guanine. Similarly, pyrimidine base has also two types, thymine and cytosine. These four bases determine four nucleotides, which are the genetic alphabets. A purine base, adenine, joins with a specific pyrimidine base, thymine only, and guanine combines with cytosine to constitute the rungs of DNA ladder. These nucleotides together constitute a genetic word called amino acid. There are 20 types of amino acids. These words combine to form protein, which is a genetic sentence or a gene. The genes function by sending chemical messages to all parts of the body to influence body growth and the infinite complex of biochemical functions. A genome is the entire DNA contained in an organism or a cell, including nuclear and mitochondrial DNA. The human genome is the total collection of genes in a human being contained in the human chromosome, composed of over 3 billion nucleotides. In April 2003, the Human Genome Project was able to sequence the entire DNA in the human genome to find out that human genome is composed of around 20,000 protein coding genes. In addition to nuclear DNA, humans have mitochondrial DNA that encodes for 62 genes. Mitochondrial DNA are inherited from mother only, and each DNA is frequently used to trace maternal lines of descent. Gene and allele. Different forms of a gene are known by the term allele. The genes having two forms of alleles, or also known as allelomorphs, are known as diallelic gene. One of these two alleles may be dominant and the other recessive, or may be codominant. Recessive alleles expressed only in homozygous genotype, that is similar combination of the alleles, and failure in the synthesis of skin pigment cause a case albinism. The allele for normal skin color is dominant over the allele of albinism. Genotype of normal skin color may be homozygous, capital letter AA, or heterozygous, capital letter A and small letter a, while the genotype of albinism is homozygous, small a, small a. Similarly, the allele of wet cerumen, that is earwax, is dominant over the allele of dry earwax. Multiple allelism and polygenic inheritance. If a gene has more than two allelomorphs, it is called multiple allelism. The gene for ABO blood group system has three main alleles represented by capital A, B and O. The allele O is recessive to both capital A and B alleles, while the latter two are codominant. Alleles are said to be codominant when both of them are expressed together. The genotypes of the four main blood groups are capital A, and capital A and O for A blood group, BB, BO for B group, AB for AB group, and OO for O blood group. There are a number of traits which are not determined by a single gene but by two or more genes together. Such traits show a wide range of continuous variation. Skin color and stature are good examples of such inheritance. Such characteristics, which are determined by at least two or more genes together are known as polygenic inheritance. For instance, if two genes A and B with their respective allelomorphs, small a and small b, together determine stature, then we will have 9 genotypes with 5 continuously variable phenotypes. Sex-linked genes. The sex chromosomes, in addition to the genes for sex development, also carry a number of genes which are not related to sex determination. Such non-sex determining genes carried on the sex chromosomes are known as sex-linked genes. Thus, X-linked genes are found on the X chromosome, and X-linked genes may also be classified into dominant and recessive. Recessive X-linked disorders are rarely seen in females and affect males more frequently. This is because males have hemizygous sex chromosome combination of X and Y. That is combination of dissimilar chromosome. There is no complementary genes to control or influence over the genes present in the X chromosome. So, the gene on the X can easily express freely regardless of recessive or dominant. In females, X-linked recessive traits are expressed only when the genotypic combination is homozygous. Y-link. Since Y chromosome is found only in males, Y-linked traits are passed from fathers to son only. The genes on Y chromosome are also known as Hollenberg genes. The testis determining factor, which is located on the Y chromosome, determines the maleness of individuals. Besides the maleness inherited in the Y chromosome, no other convincing example has so far been found. Of course, hypertrichosis on the ear, also known as hairy penny, is once considered to be Y-linked, but it is yet to be accepted by all. Sex-limited and sex-controlled traits. Some genes are sex-limited in their effect, that they are expressed phenotypically in one sex only, despite their presence in both sexes. In men, sex-limited male characters such as type of beard growth, mustache, amount and distribution of body hair depend on genes common to both sexes. This is indicated by the results of interracial matings. Caucasian males on the average are more hairy than Mongoloids, but the sons from marriages between the members of the two races are often intermediate in their hairiness. This proves that the mother has contributed genes which find their expression in the male sex only. When a genotype is expressed in both sexes, but in a different manner, it is known as sex-controlled. For example, both male and female may be affected by baldness, but relatively high frequency of affected males is notable. This implies that the genes for baldness affect more easily in the males than in the females. Sex limitation is only the extreme example of control of the expression of certain genotypes by sex. Inactivated X. All the genes present in X chromosomes but absent in Y chromosome will be double dose in females and single dose in males. The problem of unequal doses seems to be solved by a mechanism that differently condenses one of the two X chromosomes in the female. That means only a single X chromosome is active in the cell of females. The genetically inactive and condensed X chromosome turns into chromatinized body, also known as bare body. The X chromosome received from the mother may be the condensed one in some cells, whereas in some cells it may be the one received from the father. This is known as Lyon's hypothesis. Stability of number and structure of chromosome. Number and structure of chromosomes in an organism are unchangeable. Occasionally, changes do occur, which are defined as mutation. Such changes, however, results into abnormal developments in majority of the cases. For example, there are individuals with sex chromosome complement of XXY or even XXXY, etc., instead of XY of normal males. These individuals have 47 or 48 chromosomes instead of 46 in normal men. Such persons develop as effeminate men with enlarged breasts, small testes, and feminine behavior. This is known as Klinefelter syndrome, a disorder in males caused by excess number of X chromosomes. Inactivated X or bare body is seen in such persons. And there is another case of less number of chromosome, that is Turner syndrome, whose chromosome complement is XO instead of either XX or XY. So such persons' chromosome number is 45 only. These individuals are phenotypically female but imperfect, who will never attain full normal size. They have childish voice and they are sterile due to underdeveloped ovaries. An extra chromosome in chromosome number 21, that is trisomic 21, causes an abnormal condition known as Down syndrome. The total number of chromosomes is 47. Such persons have mongoloid appearance with open-mouthed and stubby hands and feet. They are mentally retarded. Distortion of the normal chromosome structure also causes abnormalities. Cry-du-Chat syndrome is a disorder caused from a deletion of the short arm of chromosome number 5. This deletion results in a phenotype of mental retardation, behavioral problems, and a cat-like call. Similarly, a deletion of chromosome number 22, also known as Philadelphia chromosome, causes leukemia. Even a single mistake in genetic spelling develops abnormality. For example, sickle cell hemoglobin is resulted from substitution of amino acid glutamic acid, the DNA spelling of which is CTT or CTC, y valine, whose DNA spelling is CAT or CAC at position number 6 of hemoglobin polypeptide chain. That means replacement of a single nucleotide T in the middle of codon by A is the cause of development of defective hemoglobin S from the normal hemoglobin. Such type of minute change is called point mutation. Every single individual is genetically unique. Every single individual is genetically a new combination except the monozygotic twins, and that no two individuals are genetically similar. Even the monozygotic twins whose genetic sentences are same develop differently like two signatures of the same individual. Similarity between mother and a child is genetically only a half. So also between father and child, because a child is the product of two haploids shared by the two parents in the form of gametes, that is sperm for male and egg for female. A haploid gamete given by a parent is again a mixture of two haploids that he or she received from his or her two parents. Such mixing of genetic materials for production of gametes, that is crossing over of meiotic cell division, also known as gametogenesis, is so random that every gamete is a new combination of alleles. Eventually, every individual is genetically unique, being a new combination of two new gametes. This combination is for once only, that has never happened before and will not repeat in future. But the genes are inherited from the past generation through parents and will be handed down to the next generation. We mortal beings therefore are nothing but temporary hosts of genes. We will be perish, but the components, the genes, will continue to exist in the populations through our children. Population genetics. A population is the collection of genetically different individuals who marry among themselves and therefore share a common gene pool. Despite variation of individual level, the members of a population together constitute common characteristics because of which they are identifiable from the members of other populations. Marriage determines the boundary of a population. If this boundary is broken, new components are introduced and ultimately changes in the characters of the populations take place. Such genetic components, once introduced, are irremovable from the recipient host and become property of the latter. When two or more populations start exchanging mates frequently by breaking down the marriage boundaries, they will merge together, constituting a new population. Population genetics, in its real sense, is concerned with the genetic basis of evolution, which may be defined as the non-cyclical changes in the allele frequencies of a population's gene pool through generations. The objects of study are primarily the frequencies of genes, genotypes, and the fitness in natural populations. The term microevolution refers to such changes in gene frequencies in Mendelian populations. A Mendelian population is conceptually a breeding population, the members of whom almost exclusively marry among them. Eugenics. Eugenics deals with the application of the law of genetics for the improvement of human race. This concept was founded by Sir Francis Galton, who defined it as the study of agencies under social control that improve or impair the racial qualities of future generations, either by physically or mentally. Eugenics has had virtually no success as a social program. Two eugenic programs arose independently of each other in the last of the 19th century. The English movement was founded by Francis Galton, and it is known as positive eugenics. It had as its goal the improvement of humanity by encouraging the talented persons to bear more children. The American movement, known as negative movement, was initiated by several social reformers who erroneously identified certain related families as bearers of socially undesirable traits. In 1932, the third and last International Congress of Eugenics met in New York City, while many eugenists were still looking for defective families and ways to encourage differential reproduction, many other speakers criticized the eugenic movement for its elitism and racism, etc. Conclusion. Eugenic movements in the past might be a great failure, but the importance of genetics for better life and in solving mysteries of life is endless. Genetic counseling for making decision by prospective partner about their genetic compatibility and incompatibility, or about taking decision whether to have a child or not by the couples who carry serious hereditary defects, diagnosis of genetic diseases and treatment thereon, identification of certain genes that have adaptive or protective advantage against certain diseases, and identification of individuals for forensic purposes, tracing of human origin and diversity, etc., are only a few of its wide applied value. That's all about human genetics. Thank you.